Hi, welcome to Biology 5 Online Lab. For our first lab, we're going to be going over the microscope as well as important lab safety information. While you may not need this information right away, since we will not be handling microscopes in person or working in an in-person lab, it's important to note this information for future biology classes you might be taking. Let's get into why we need microscopes in the first place. When we look at the living world, we can really divide it into two major sections. The macroscopic parts, which are the large scale things that you can see with the naked eye, and the microscopic parts, where things are too teeny tiny for you to see with the naked eye. When something is microscopic, we need some sort of tool to enhance our ability to see that organism or that piece or cell or whatever it is we're looking at. Microscopes are the main tool that we use to do that. Scientists will use these microscopes to observe organisms that are smaller than what the naked eye can see. Mainly, we'll be looking at the light microscope. A light microscope is any microscope that uses the visible light spectrum to illuminate a sample. So what can we see under a microscope? We can see various samples that are usually related to a small living component. Sometimes, though, it's also a piece of a larger organism, or sometimes they're single-celled creatures. We could see something like bacteria or a protist, which are single-celled organisms where we can see the entire organism under the microscope, or they can be those pieces of a larger organism, such as a sample from our own human skin, or from a liver, or from a lung. There's two types of creatures that we would look at: eukaryotic creatures and prokaryotic creatures. Prokaryotic cells are things like bacteria that are single-celled, and eukaryotic cells are multicellular for the most part, where they have lots of organelles and components, and then we're thinking of things like animals and plants and protists. We'll get more into the cells in another lab. When we're looking under a microscope, especially when we're looking at something that is larger than what could fit under the microscope, like a tissue sample, we see them cut into pieces. We're seeing a piece, and there's a few terms we can use to describe how something is cut. There's the cross-section, the longitudinal section, and the hole mount. We'll start with the hole mount since that one is a little bit more self-explanatory. The hole mount is the whole organism. So if you take a sample of bacteria and you see all of the bacteria under the microscope, that would be a hole mount. Cross sections and longitudinal sections are better explained by looking at this apple. The longitudinal section runs from the top all the way to the bottom of our apple, whereas the cross section is a section taken from the middle. So in the longitudinal section, we see the entire core from the stem all the way down to the bottom here. And when we take the cross section, we see the entire round part of the core, but we don't see the stem and we don't see the bottom. We're just getting a sliver in the middle. So if you think about yourself and what would be longitudinal or cross section for yourself, longitudinal would be if you cut yourself from your head all the way down to your toes, whereas a cross section would be like if you took a slice right out of the middle. Now, we mentioned light microscopes, and we said that there were other types as well. We're going to focus on the light microscope, but let's introduce the idea of the electron microscope as well. And there's some pretty big differences between these. The light microscope is what we see here. And so this is probably what you're going to use for the most part in your life science career, is a microscope that looks something like this, where you have some pieces for you to look through, some various magnifying pieces on the inside and the objectives, the light source coming from the bottom and illuminating whatever you are looking at that would be held here on the stage. An electron microscope is very different. Instead of using visible light, we're going to be shooting a beam of electrons at whatever it is we're trying to look at. And so you have a much larger piece of machinery involved for that process, and the images are seen on a computer instead. If you don't need to use an electron microscope, you probably will never use one. So we're going to dive into the light microscope and really leave the electron microscope off to the wayside. Now there's two different light microscopes you might use, the stereoscope and the compound scope. The stereoscope, also called a dissecting scope, allows for you to see an entire organism in 3D. It only uses one lens, which is why we call it a stereoscope. And this is only good for low magnification. So if you wanted to look at what a cricket's face looked like, the stereoscope would be a great tool. But let's say you want to see something smaller. That's where the compound microscope comes in, the compound light microscope. And that's any microscope that uses two or more lenses to magnify our specimen. So in this compound light microscope we see here, there's two lenses that are going to help magnify whatever it is we're going to look at. So we can see things that are smaller, but we do lose a little bit of that 3D nature. Things have to be much smaller and flatter and thinner for us to be able to view that specimen. Let's look at some of the parts of the compound light microscope, since this is the one that you'll probably use the most. And we're not going to get into every single piece in this microscope, but we're going to get into a few. So the ocular lenses, or the eyepieces, is where you're going to look through. Right? And you see it kind of looks like a pair of binoculars attached to the rest of the contraption. The eyepieces actually do magnify. They do have a lens in there that can magnify whatever it is you're looking at. We have the head that connects the ocular lenses to the rest of the microscope. We have the arm, which extends down, the base, which is the bottom. We have a light source, which provides the illumination, an on and off switch, a knob that can help adjust the brightness of our light source, a condenser, along with the stage where we will hold on to our sample that we're viewing, the objective lenses, of which there are a few, which are rotated around by a nose piece. These objective lenses provide more magnification, and it provides adjustable magnification. So depending on what you're looking at, you may want to magnify it only 40 times larger than what it really is. Or if it's something tinier, you might want to magnify it 400 times more than how it looks in real life. By rotating this nose piece, you can select the appropriate objective that will magnify your sample at whatever magnification you're looking for. We also have some knobs and controls down over here. We have a stage control, which will move the stage forwards and backwards and left and right. That's extremely important if you're trying to find something on your slide. Maybe you're looking for a particular type of cell, or maybe you have some living cells on your slide and you're trying to follow them as they move. You can use the stage control to do that. And then we have the coarse adjustment knob and the fine adjustment knob. Now when you put your slide on the stage, it's not going to be perfectly focused. You're going to have to bring it into focus. You can do so by using the coarse and fine adjustment knobs over here on the sides. These knobs both will do the same thing. They will move this stage up and down to help you bring the image into focus. The difference between the two is that the coarse adjustment knob will make really large changes to how high or low the stage is, whereas the fine adjustment knob will make very subtle changes. So when you're first putting your slide on the stage and you're at a low magnification, trying to find the image, trying to get things relatively in focus, the coarse adjustment knob is a great place to start. However, once you have a rough idea what you're looking at, then you want to use the fine adjustment knob. So the coarse adjustment knob is great for finding the initial picture you're looking for. The fine adjustment knob will fine-tune to make it a nice, crisp, clear, sharp image for you to look at. The coarse adjustment knob should only be used when you're at a low objective, something like 4x or 10x. It should never be used on a high-powered objective. The reason being is that you'll easily lose your picture since the magnification is so high, and you also run the risk of damaging the equipment since you might accidentally crash the stage up into the objective and scratch those lenses. Once you've found your picture on one of those lower objectives, you want to only use the fine adjustment from there on out. Once you go to a high objective, you should never use the coarse adjustment and only use the fine adjustment for any little changes you might need to make. When you're focusing the microscope, you'll notice that when you're using different objectives, you are doing a kind of a trade-off. So here we have a total magnification of 40x. Usually the ocular lens is 10 and the lowest objective called scanning is 4. 10 times 4 is 40, so the total magnification would be 40 on the lowest objective. What we see as we go up in the objectives and we increase the magnification, our field of view becomes smaller. We're able to see the object as larger when we're at a higher magnification, but we don't see nearly as much around it. So everything that was kind of on the peripheral out here on the 40x is going to disappear when we look at it in 400 because it's somewhere off to the side outside of our field of view. We also lose depth as we increase the magnification. So when you look at something like threads, like this red, yellow, and blue threads, when you look at it the 40x, all of the threads may appear to look somewhat in focus. But as you go up to the 100 and the 400x magnifications, you'll notice that only one thread can be in focus at a time. Because as we increase magnification, our depth of field goes down. They're inversely related to each other. So some of the things you'll be doing in your lab, and of course you will not have a microscope at home, so you will be either looking at a drawing, watching a video, or viewing a prepared slide already. But you'll be looking at a letter E, you'll be watching a video on threads, and you'll also look at an image of a human blood smear. So you'll be looking at all of these in your first lab. When it comes to drawing what you see, and this is very important as we go through this lab, you'll notice in your lab that you have the space that's given for you to draw different samples. All of the samples will be available either in the lab or in an accompanying lab guide. But when you're drawing, you want to make sure you're including the following information. You want to make sure you give it a title and that the title makes sense. What are you looking at? Maybe identify where the specimen, what the specimen is, or where it came from. For example, you could put simple squamous epithelial tissue, or you could say lung tissue sample for a title. For the field of view, our field of view is this circle here. You want to make sure you draw everything you can see in your field of view in its entirety and try to be accurate with size. Now, I understand that that will be a little bit difficult since we are doing this online and using images, but do it to the best of your ability. It's safe to assume that a cell is not going to take up the entire field of view. You're probably going to be able to fill many cells into the space. Also, make sure to include labels. Label things that are important or that you're instructed to label. Something like the plasma or cell membrane, for example here, and the nucleus. You can label things by drawing a line from the object that you're identifying to the outside of the field of view, and then writing in what it is. It's better to over-label than under-label, so if you're not sure, play it safe and give it a label. You also want to include the magnification. In some spots you'll be given a space for this, but if you're not given a space, you could always include it in the title. We want to include total magnification, which is the ocular times the objective magnification. For the sake of this course, assume that the ocular is times 10, and then the objective is 40. For most things you're looking at, you should have a 400x, as most things are going to be magnified at a very high objective. Moving on from microscope work, we get into lab safety. Again, we're going to be at home. That's the safest thing that we can do right now. However, in future classes, lab safety is paramount. The procedures and policies in your lab classes are made to keep you and your classmates safe. So when you're in a lab setting, make sure you're always following your lab protocol, any directions that your instructor gives you, as well as common sense. If something doesn't seem safe, don't do it, or at least ask. And that's really our golden rule when it comes to lab safety. If you ever have questions, or you do not know what to do, or where to put something, or where something goes, ask your instructor. Your instructor will be happy to tell you what you need to do, or where something goes. We would rather you ask and avoid an injury, or a spill, or something along those lines, rather than you just trying to go off and do it on your own. We'll never berate you, we'll never look at you as silly or uninformed because you're asking a question when it comes to safety. And another key thing too, if you see someone doing something unsafe, say something. Everyone is in charge of safety in the lab classroom. If you see something, say something. When it comes to labs, there's proper attire when it comes to safety. And depending on what the procedures are or what materials you're using in the lab, that attire can be a little bit different. But in general, most labs require the following: long pants or the equivalent, closed-toed shoes, and clothing that is not excessively torn or exposes a lot of skin. The reason being is that when we're dealing with sharp objects and things that can contaminate us, having clothing to protect our skin is going to be more helpful. If something were to spill on you that was dangerous, it's a lot easier to remove the clothing, and with the clothing comes the contamination, than to try to fix the contamination directly on your skin. So it's not because we want to be prudes or anything, or control what you wear. It's just we want you to be safe. Depending on what the lab is, or what procedures are being done, you may also have to wear a lab coat, eye protection, or gloves as well. It's important to know what you're doing in lab that day so you can come prepared. And if you have questions on lab attire, again, ask your instructor. Food and drink in the lab. It's really easy to accidentally contaminate things in a lab. It's easy to accidentally touch things that we shouldn't have touched and then go back and touch other things that we shouldn't have touched. So food and drink is generally never allowed. If you do have food or drink, you should pack it away in your bag and keep it out of sight. Really try to avoid something that cannot seal. So things like a Starbucks cup that has a straw wouldn't be a good thing to bring into the lab since you can't actually seal it and properly pack it away. Sometimes labs do go long though, and you need a snack or a break, and that's okay. Make sure to talk with your instructor about what's allowed or not when it comes to breaks. But in general, if you're hungry or thirsty and you really do need that break, just make sure that you wash up, make sure that your lab bench is safe and clean, wash your hands, take your snack outside of the lab, enjoy, and then come back in. Don't consume any sort of food or drink without washing your hands, and don't consume it inside the lab itself. Proper disposal is also very important. And again, follow the instructions of your instructor or your lab protocol when it comes to proper disposal. There are a lot of substances that cannot go in the regular trash can or down the drain. Many things that we deal with on our day-to-day life can just be thrown into the garbage or poured down a drain, and that's fine. And in the lab, sometimes that will be the case. Paper towels can go in the garbage, water can go down the drain, and that's fine. But there's a lot of things we use in the lab that cannot go in the trash or in the drain. Biological components, such as things from dissection. If you are cutting up a liver or a fetal pig, you can't throw those biological components in the regular garbage. Instead, they'll go into a biohazard bag that will be specially dealt with before being handed off. Harmful chemicals cannot go down the drain. Harmful chemicals will be collected in specially marked liquid waste collection containers. It's important to note when you're in the lab. Sometimes if you're dealing with more than one chemical, you may have more than one liquid waste collection container. Make sure to read the label that you're disposing the right thing in the right container. If you're using needles or glass slides for a live or a wet mount, when you're done with those, they go into a sharps container. This is usually a red or sometimes white container that has a biohazard logo on it, and it is a special form of biohazard disposal for things that are sharp, as the name implies. Does not include broken glass though, so only needles and intact glass can go in the sharps container. Broken glass has its own specific container, usually a cardboard container marked broken glass. Do not ever put broken glass in the regular trash can. You run the risk of injuring those who help keep your lab nice and neat and tidy if you do. Just in case something does go wrong, because even if you take all these precautions, sometimes people still get injured or hurt, in which case we have lots of safety equipment in the lab to help prevent any injuries from getting worse. We have a first aid kit that will have things like bandages and gauze, as well as antibiotic ointments. There's always an eye wash station for you to flush your eyes if something were to splash in your eyes or on your face. There's an emergency shower if something were to spill on your body, where you could pull the handle and water will pour down in the lab. There's also usually a fire extinguisher or a fire blanket. It's important to note with fire extinguishers, if you don't know how to accurately use one, please don't try. Instead, just call for the fire department. But if you do know how to use a fire extinguisher, there's usually one at your disposal if there is a fire. There are also spill kits in case certain chemicals are spilled onto the floor or tables. And lastly, a telephone. When in doubt, call for help. Right? Your instructor should be informed on how to handle most, if not all, of these problems in the lab. But if it's something beyond you, your classmates, and your instructor's control, then the telephone is a wonderful tool to use in order to get someone out of the room, to have some sort of hazard taken care of, or to get someone help or treatment that they might need in the case of a very severe accident. Accidents are generally rare, and I don't expect any of you to ever have to use any of this equipment. But even though it's a very rare thing to happen, you still want to be aware of where these items are in the labs that you are in.